Hello everyone, I'm Sonali, Assistant Professor in Botany, presenting to you this video lecture on the topic Reproductive Structures of Pinus as part of an initiative by DHE, Haryana. This lecture will cover the topics: first, general introduction to Pinus reproductive structures, followed by morphology of the male reproductive structure of Pinus and how male gametophyte and male gametes are formed and develop. Next, it will discuss the morphology of female reproductive structures of Pinus and how female gametophyte and egg cells are formed and develop. And at last, it will talk about pollination in Pinus. So, let us see what are the reproductive structures of Pinus in general. The tree is monoecious, that is, it bears both male and female sex organs, but those organs are born separately on different cones or structures. We will start from the most basic structure for reproduction, that is gamete. In Pinus, male and female gametes are formed inside a special structure known as gametophyte. To be precise, male gametophyte and female gametophyte. This gametophyte is developed from the structure known as spore. The development is endosporic, that is, gametophyte develops inside the spore, does not break out of the spore wall. Now, depending upon the sex, male gametophyte develops from microspore and female gametophyte develops from megaspore. Now again, microspore are formed inside special structures called microsporangia and megaspore from megasporangia. These sporangia are borne on special leaves modified to bear reproductive structures known as sporophylls. Again, they are of two types, microsporophylls and megasporophylls. These sporophylls are arranged spirally on a central axis and form structure known as cone. They are compact, hard and woody structures borne in the axils of scale leaves and are dioecious in nature. A structure similar to cone is found in other gymnosperms and that is known as strobili, but the difference between strobili and a cone is a tightly compactly arranged structure, whereas strobili is a loosely arranged structure. So, let's start with the Pinus male cone. The picture you are seeing is of Pinus roxburghi species bearing lots of male cones. Male cones are small structures, arise in the lower branches from around January to March in clusters of up to 140 cones in the axils of scale leaves in place of where dwarf shoots were to be born. They are born in apical region of young newly formed stems or branches. An involucre of bracts is present at the base of each cone. In the picture, you can see an old cluster of cones, brownish in color, and behind that is a young cluster of male cones, smaller and green in color. When developing, the cone is compact and closed, not exposing microsporangia, but at the time of pollination, the central axis of cone increases in length, making sporophylls less compactly arranged and exposing microsporangia to the outer environment for releasing pollens. Now, This is a longitudinal section of a male cone. There is a central axis, and on that 60 to 100 microsporophylls are arranged. Upper and lower sporophylls are less fertile. On the lower or abaxial side of microsporophylls, 2 microsporangias are formed. Microsporangia are sac-like structures in which microspores are formed. In the diagram on right side, you can see a closer look at microsporophylls and microsporangia. Microsporophyll has two parts: fertile proximal part towards the central axis and distal sterile part, terminal. This terminal part is known as apophysis and is flat, pointed, scale-like structure. It is woody. On the lower surface of sporophylls are borne microsporangium. Inside them are yellow-colored winged pollens. After the structure, we now move on to the different stages of how these structures are formed. One thing we need to remember is that the Pinus follow eusporangiate type of development. That is, the gametophyte develops from a group of initials or cells instead of a single cell initial. So, what happens is, on the abaxial or lower surface of microsporophyll, a small protuberance develop from a group of initials. Outermost epidermis layer forms outer covering, and inner hypodermis cells enlarge in size and transform into archisporial cell. Archisporial cell now divides into a primary parietal cell on outer side and a sporogenous cell on inner side. Parietal cell undergoes a series of periclinial and anticlinial cell divisions, forming 2 to 3 middle layers and 1 tapetal layer on the innermost side. So now we have outer epidermis, 2 to 3 middle layers and 1 inner tapetum layer as different layers of microsporangium. Here, the function of tapetum is similar as that of tapetums in angiosperm, that is, to provide nutrition to developing microspores. Also, sporopollenin is secreted by tapetum, which gets deposited on the surface of pollen grains. Now, on the other hand, sporogenous cell divides in all planes, forming a mass of cell. These cells act as microspore mother cells. Each microspore mother cell undergo meiosis and form tetrad structure of 4 microspores bound to each other by callose. After dissolution of callose, 4 microspores get transformed into 4 pollen grains. They are winged structure. Later on, outer epidermis develops fibrous thickenings in all of its cells except for few. This will help in dehiscence of microsporangia at the time of pollination. Now, next step is microsporogenesis, that is, development in microspore or pollen grain. The nucleus of pollen grain undergoes division and form a small flat prothelial cell on upper side, which is ephemeral in nature, that is, it degenerates later, and a large central cell. Initially, the two nuclei are separated by callose deposition. Cellulosic wall develops later in between these two nuclei, forming two cells. Now, the central cell again gives off second prothelial cell and one enthedial initial. The enthedial initial divides to form one small enthedial cell and one large tube cell. Now, at this stage, pollination will take place and the pollen has two prothelial cells which degenerate later, one enthedial cell and one large tube cell. After pollination, enthedial cell divides into stalk cell and body cell further divides into two male gametes. Now here we have a closer picture of winged pollen grains of Pinus. In the diagram you can see outermost layer is exine and is restricted to one region. Next is middle layer, meseine. This layer extends laterally into sac or balloon-like structures. They are initially filled with fluid, but by the time pollens are released, the fluid gets dried up, leaving empty space, and this empty space forms sac, which helps in dispersal through the wind like wings. Inner layer is a very thin intine which encloses prothelial and enthedial cell. Now we will move on to Pinus female cone. This is a picture of female cone of Pinus sylvestris. They are borne on upper branches. If you remember, male cones born on lower branches. So, female cones born on upper branches in the axils of scale leaves in place where long branches were to be born, and in cluster of 2 to 4 cones. Each cone consists of 50 to 60 Megasporophylls arranged spirally on a central axis. They take 3 years to mature. And here we can see 3 pictures of female cone at different stages. First is the young female cone, just born, just arise or in the axils of scale leaves. Next 2 images are of developing cone after 1 year and then ripened one after 2 years. It is around this stage that pollination takes place, and the 3-year-old female cone is the one which has elongated central axis with exposed megasporangia and has started seed formation. It is similar to the structure one you have seen in the previous image. Now this is a longitudinal section of a mature female cone. Upper and lower sporophylls are less fertile. Megasporophylls are made of 2 parts: fertile ovuliferous scale and sterile bracт scale. First, bracт scale is large in size, but later on, as the cone grows and increases in size, ovuliferous scale becomes larger than bracт scale and bear 2 megasporangia on the upper or abaxial surface. The terminal part of ovuliferous scale is known as apophysis, like in Microsporophyll. In the diagram, you can see a closer view of megasporangium with ovule on upper side covered with integument except at micropyle. Now let's move on to how the female gametophyte and egg cells are formed. Here also Development of gametophyte is of eusporangiate type, and it starts with the formation of a cellular protuberance on surface of Megasporophyll. To be precise, on the upper surface of Megasporophyll. Outer integument is unitegmic and crassinucleate nucellus is formed. Hypodermal cell transform into archisporial cell. Archisporial cell divides into primary parietal cell at towards micropylar end and sporogenous cell at chalazal end. Parietal cell divides anticlinally and periclinally and form three-layered covering of outer fleshy sarcotesta, middle stony sclerotesta and inner fleshy sarcotesta. These layers cover the female gametophyte, that is megaspore. Now the sporogenous cell at chalaza end transforms into megaspore mother cell, which undergoes meiosis and form a linear tetrad of 4 megaspores. Now there is a differential movement of cytoplasmic content and starch towards the megaspore at chalazal end. The upper 3 megaspores towards the micropyle degenerates and the one at chalazal end becomes functional megaspore. It has high starch and cytoplasmic content. Next is free nuclear division that takes place in functional megaspore with no wall formation. Size of megaspore also increases and later slowly callose wall is laid down followed by cellulosic wall. Now 2 to 4 cells at micropylar end starts functioning as archegonial initials and form primary neck initial on outer side and a large central cell on inner side. 4 neck cells are formed from neck initials. They remain arranged in single tier or single layer. On the other hand, central cell enlarges in size, transforms into archegonium and form 2 types of cells, ventral canal cells on outer side and egg cell on inner side. Now here is a diagrammatic representation of few major stages in development of egg cell. In first figure you can see yellow colored nucellus enclosed in unitegmic integument and megaspore mother cell inside. This structure is born on ovuliferous scale. Next is formation of 4 megaspores by meiosis, 3 of which degenerates in next figure and the one at chalazal end becomes functional megaspore. In the next figure you can clearly see nucellus tissue slightly separated from archegonium. At the end, some archegonial cells around the egg cell divide and grow rapidly so that egg cell sinks inside forming a space. This is known as archegonial chamber. And now here are pictures of female Pinus cones of species that are found in India. You can clearly see how they differ from each other depending upon their size, shape, number of sporophylls and structure. A is female cone of Pinus gerardiana. It is a large cone. B is cone of Pinus wallaceiana species and the cone is narrow and elongated. C is Pinus insularis female cone with a smaller cone and less number of Megasporophylls. D is Pinus aramendi species, which again is a long cone and broader in diameter but shorter than species wallaceiana. And at last, E. Pinus roxburghi. The cone is of medium size. Now, coming on to the last topic of this lecture, pollination. Pinus uses wind as pollinating agent. A huge number of pollens are formed to ensure fertilization. For pollination, the epidermal wall with fibrous thickenings shrink. This dehisces microsporangium like a burst and winged pollens are released. The color of pollens is yellow, which is why when released in large number it looks like sulfur dust has been released in air because of yellow color. This is also known as sulfur shower or golden shower. These pollens are then trapped in mucilaginous drop at micropyle secreted by nucellus of ovule. Later, the mucilage drop dries and seal the micropyle so that no more pollens can enter. So with this, we come to an end of our lecture. And I would leave you with another small exercise where you have to find out what is megasporogenesis and what is Pinus male gametophyte. Hope you have learned something from this lecture. Thank you for watching.